Okay, here this week lecture starts with the discussing about the stabilizing residues. So we'll start this session discussing what are stabilizing residues. Stabilizing residues of a protein are amino acids that play critical role in maintaining the stability and conformation of a protein. So why this stability is needed? So this stability is needed for the functional reasons. So these stabilizing residues are usually located in the core of the protein and are involved in stabilizing the protein secondary structure, tertiary structure and quaternary structure through various interactions. So here I will be discussing some of the stabilizing interactions that proteins have. The first is hydrophobic residues. So hydrophobic residues include leucine, valine, isoleucine and other non-polar ones. So when we see a folded structure, so these hydrophobic residues are located in interior of a protein where they form hydrophobic interactions with other nonpolar residues. So these interactions are essential for maintaining the stability of protein structure. Next is the salt bridges. As you know, these are formed between two oppositely charged residues, such as positively charged amino acids or negatively charged amino acids in between them. So these interactions are crucial for maintaining stability of a protein structure by helping to stabilize the charged regions of a protein. So these charged regions mostly will be the exposed or the surface regions. Next will be disulfide bonds. As we all know, disulfide bonds are formed between two sulfur atoms like cysteine residues. So these disulfide bonds play a critical role in stabilizing the tertiary structure of the protein by helping to hold together specific regions of the protein structure. Finally, hydrogen bonds. So these hydrogen bonding interactions helps to stabilize the secondary structure of the protein such as alpha helices and beta sheets. To identify the stabilizing residues in a protein, computational methods such as molecular dynamic simulation or free energy calculations can be used. These methods can help to predict the interaction between the amino acid residues and provide insight into the stability of protein structure. In summary, the stabilizing residues of a protein are essential for maintaining the stability and conformation of the protein structure. These residues interact with the other residues through hydrophobic interactions, salt bridges, disulfide bonds, and hydrogen bonds to stabilize the protein structure. Computational methods can be used to predict and identify these residues in a protein. Predicting the stabilizing residues in a protein is an important task in protein engineering and drug design. Several approaches have been proposed to identify these residues based on their physical and chemical properties. Various computational methods can be developed to predict these stabilizing residues based on their physical or chemical properties. For example, machine learning algorithms can be trained on a known stabilizing residues to identify the patterns in some properties. Okay, for example, surrounding hydrophobicity or long range order or stabilizing center or conserved residues. So observing the pattern or cutoff values of this we can develop a machine learning algorithms that can be trained to predict the stabilizing residues. Okay, here I'll be discussing some of them. First, let's start with the surrounding hydrophobicity. So this approach is based on the idea that hydrophobic residues are often located in protein core and play an important role in stabilizing the protein structure. This approach involves the calculation of hydrophobicity of amino acid residues surrounding for a given residue and identifying residues with high hydrophobicity values as potentially residues. So I'm repeating, in this approach, First, we calculate the hydrophobicity of a residue based on the based or sum of the uh, hydrophobicity values of the surrounding residues. Then, once we calculate this surrounding hydrophobicity for all the residues, is the residues with high hydrophobicity values are potentially termed as stabilizing residues. And when we see these residues with high hydrophobicity are more likely to be buried in the protein core and interact with the other hydrophobic residues, contributing to the overall protein structure. Next is the long range order. So this is based on the idea that protein are often determined by long range interactions that residues far apart in the protein sequence. So this approach involves analyzing the sequence and structure of a protein to identify the residues that are spatially close and have similar physiochemical properties. Residues that are involved in long range interactions are often highly conserved and can be predicted as stabilizing residues. Next is the conservation score. So this approach involves analyzing the sequence of a protein and identifying the residues that are conserved across the set of related sequences. So how can we observe the conserved sequences? So we can do multiple sequence alignment, obtain a multiple sequence alignment of a set of proteins, then we can see conserved and non-conserved part. So here, residues that are highly conserved are more likely to be involved in stabilizing the protein structure and can be predicted as stabilizing residues. Similarly, residues that are involved in long-range interactions are often highly conserved and can be termed as stabilizing residues as well. Next is the stabilization center. This approach involves the identifying regions of the protein that are important for stabilizing the protein structure and analyzing the amino acid residues in this region to identify stabilizing residues. So these stabilization centers can be identified using experimental methods such as mutagenesis or computational methods such as free energy calculations or molecular dynamic simulations. In summary, predicting the stabilizing residues in protein involves analyzing the physical and chemical properties of amino acids and identifying the residues that are likely to play a role in stabilizing the protein structure. Approaches such as surrounding hydrophobicity, long range order, conservation score, and stabilization center can be used to identify the stabilizing residues in proteins. These approaches can help or guide in protein engineering and drug design efforts by identifying key residues that can be targeted for modification or optimization. Okay, here I have some basics question on so far that we have discussed. So the first question is, residues with what hydrophobicity are more likely to be buried in the protein core and interact with what type of residues contributing to the overall residue. Here I have first question for today. So you can answer using the chat box. So residues with what hydrophobicity are more likely to be buried in the protein core and interact with what residues contributing to the overall stability of the protein structure. Yes, I started receiving the answers. Everyone, please mute yourself. Yes. So residues with high hydrophobicity are more likely to be buried in the protein core. So we call it hydrophobic collapse, also other name for this. Then they interact with the other hydrophobic residues, contributing to the overall stability of protein structure. So second question is residues that are uh, what conservation level are more likely to be involved in stabilizing the protein structure and can be predicted as stabilizing residues. Yes. Yeah, residues with high conservation are more likely to be involved. So I want everyone to be involved in answering so that that creates an environment for a good discussion. Even if it's right or wrong, just let me know your answers. Okay, moving on from here. So I'll be discussing detailedly the surrounding hydrophobicity followed by long range order. So this surrounding of hydrophobicity is a computational method used to identify the stabilizing residues in proteins based on their hydrophobicity and location in protein structure. This approach involves calculating the hydrophobicity of amino acid residues surrounding a given residue and identify residues with high hydrophobicity values as potentially stabilizing. The hydrophobicity of an amino acid residue is typically measured using a hydrophobicity scale, which assigns a numerical value to each amino acids based on its tendency to be located in hydrophobic environment. One common method or scale used in hydrophobicity scale Doolittle scale, which assigns a numerical value to each amino acid based on its free energy of transfer from non-polar solvent to water. So here the formula for calculating the surrounding hydrophobicity is sum of hydrophobicity values of a residues within a given distance from a residue I divided by number of residues within the given distance. For example, if we want to calculate the surrounding hydrophobicity of a residue I using a distance of 5 residues, think 5 residues are within the distance cutoff, we would sum the hydrophobicity values of 5 residues divided by 5 number of residues surrounding. So these hydrophobicity values of the residues are taken from the scales, that Kyle-Doolittle scale. Here, residues with high surrounding hydrophobicity values are more likely to be present in the core and interact with the other hydrophobic residues, contributing to the stability of the protein. Next is the long range order. This is also a computational method used for identifying the stabilizing residues based on the long range interactions between the residues that are far apart in sequence. This means that this takes into consideration residues which are far apart in sequence. So for example, they take with the residues that are 12 residues apart has far residues, and in the structure they are nearby. So this approach involves analyzing the sequence as well as structure of a protein to identify the residues that are spatially close and have similar physiochemical properties. Residues that are involved in long range interactions are often highly conserved and can be predicted as stabilizing residues. One way to quantify the long range order is to use the mutual information score between the pair of residues in protein sequence. So the Mi score is a measurement of degree of coevolution between two residues, and it can be used to identify residues that are likely to interact with each other. Here, residues with high Mi scores are more likely to be involved in long range interactions and can be predicted as stabilizing residues. Other methods that can be used to identify the long range interactions include coevolution analysis, contact prediction, and network analysis. Okay, moving for the preferred residues or the location and preferential observation of these residues. So stabilizing residues in protein can be classified into different categories based on their location and function in protein structure. The preferred stabilizing residues and their location depends on specific protein and its structural features. So here I will be discussing some examples of stabilizing residues and their location. First, let me start with the core residues. These are highly hydrophobic residues located in the protein interior, where they form a tightly packed core that stabilizes the protein structure. Core residues are often highly conserved and can be predicted using methods such as surrounding hydrophobicity approach. Next is the surface residues. So mostly surface residues are charged or polar, located on the surface of the protein, where they participate in electrostatic or hydrogen bonding interactions that stabilize the protein structure. Surface residues can be predicted using methods such as uh, electrostatic potential analysis or conservation analysis of the surface residues. Next is the interface residues. So what is difference between an interface and a surface is, interface is a subset of surface. So when two proteins interact with each other, the interaction part or where they bind is called as a interface. So these interface residues are located between protein subunits or between protein and its ligand, where they form an interactions that stabilize these complexes. So these interface residues can be uh, predicted using methods such as contact prediction or interface conservation analysis. Even if we know the complex structure, these interface residues can be predicted by comparing the accessible surface area and distance-based methods as well, which during the course we will be learning those methods as well. Next is the disulfide bonds. So these stabilize the protein structure by covalently linking two parts of a protein. So these disulfide bonds can be predicted using methods such as conservation analysis of cysteine residues or prediction of disulfide bonding patterns. In general, stabilizing residues are often located in regions of a protein that are important for its function or stability, such as in active sites, in binding sites, or in structural motifs. The specific locations of stabilizing residues can vary depending on protein and its structural features, and different methods can be used to predict them. Okay, moving forward, we see how the location or in a class of proteins. Till now, we have not preferred stabilizing residues are amino acid residues that are commonly found in cores or interfaces and are important for maintaining the stability and structure of a protein. So these residues are typically hydrophobic and have high propensity to form stabilizing interactions with each other residues such as van der Waals interactions, hydrogen bonds, and salt bridges. The location of preferred stabilizing residues depends on the structure and function of a protein. So in globular proteins, preferred stabilizing residues are typically located in protein core, where they form hydrophobic interactions with other hydrophobic residues and stabilize the protein's compact structure. These residues are often buried and inaccessible to solvent molecules. In contrast to this, what we have studied, surface exposed residues are often hydrophilic and charged, allowing them to interact with the water molecules and participate in protein-protein interactions. In membrane proteins, preferred stabilizing residues are often located in the transmembrane region, where they interact with the lipid molecules and other hydrophobic residues to stabilize the protein structure. These residues are typically hydrophobic and have high propensity to form alpha helices or beta sheets. That is why when you see a membrane protein and having it spans the membrane or lipid bilayer using secondary structure elements such as alpha helices and beta barrel. In protein-protein interfaces, the preferred stabilizing residues are often located at the interface between two proteins. So these residues are typically surface exposed and can be either hydrophobic or polar depending on the nature of the interface. So the identification of preferred stabilizing residues is important for understanding the structure and function of a proteins and for discerning the stable and functional proteins. So experimental methods such as X-ray, NMR can also be used to identify and validate the location of preferred stabilizing residues in a protein in combination with the metagenesis studies. Similarly, experimental methods also. Okay, next we start with the thermodynamic databases. So biological thermodynamic databases are collection of information about the thermodynamic properties of biochemical molecules such as proteins, nucleic acids, and metabolites. These databases are important for understanding the energetics of biochemical reactions and processes and for developing models of metabolic networks and uh, pathways. So let's start with the Prothom. Prothom is the largest, till date, it's the largest database which is maintained by our lab, that is Professor Michael Gromia's lab. So is one of the most comprehensive databases of proteins and their mutant information. So it contains information on Gibbs free energy, enthalpy, entropy, and entropy changes. These all thermodynamic changes associated with protein folding and stability. So this Prothom also includes experimental conditions such as temperature, pH, ionic strength, as well as structural information about the proteins. And we also have a subset database of Prothom, which is NPTherm, which is a thermodynamic database for membrane proteins. Next is the PDB-ReDO. So this is a database of refined protein structures that provides information on thermodynamic stability of proteins. It contains data on Ramachandran plot, side chain conformations, and the quality of the protein structures which can be used to predict the protein stability. Next is the STP-PRED. So this is based on a computational prediction database where machine learning algorithms are used to predict the stability of a proteins based on their sequence and structural features. It can predict the Gibbs free energy changes associated with mutations in protein and can use to identify the stabilizing mutations in protein. So in general thermodynamic studies, Gibbs free energy of unfolding or Gibbs free energy of folding that we obtain to the chemical denaturation experimental studies is considered as a stability value. So to define what is stability in a protein structure, so it is the difference between the free energy of folded and unfolded states. So from these experimental papers, delta G, which defines Gibbs free energy of unfolding, is generally considered as stability value. Just give me a second. Next is the Foldex. which is an empirical force field, which uses an empirical force field to predict the thermodynamic stability of proteins. It can be used to calculate the Gibbs free energy of folding or unfolding, as well as the effect of mutations on protein stability. The last one is protein stability database. So this has information of protein ligand interactions. So it contains information like Gibbs free energy, enthalpy, entropy changes associated with protein ligand binding, as well as experimental conditions such as temperature and pH. So these databases can be used to study the thermodynamic properties of proteins and predict the effect of mutations on protein stability and design new proteins with improved stability and function. So in the last part of the session, we'll be discussing how these different databases are used to develop the different machine learning methods. So considering their importance in current day study, these are the valuable resources for researchers studying protein structure and function, as well as for drug discovery applications. Okay, now so far we have discussed the databases. So what is the source of this data to build these databases? The first thing is experimental measures. So these experiments often include data from so the thermodynamic databases that often include data from experiments that measure the thermodynamic properties of biochemical molecules. So these experiments, so what experiments we use is techniques such as calorimetry, spectroscopy or chromatography to measure the enthalpy, entropy and Gibbs free energy of the molecules. Second is computational calculations. So these calculations can be used to predict the thermodynamic properties of molecules based on their structure and composition. So these are the prediction based. Just like we have read STP-PRED, which is a machine learning based prediction database. Database having the prediction data. Next is the literature. So most of the current day database, including Protham and other, are literature review databases. So what they do is the developers or the curators of these databases, like literature databases based on literature reviews, may review the literature and select the data that meets certain criteria, such as the accuracy of measurements and the reliability. Then they build a database. They collect the information from different publications or literature and they build. Next is the data sharing. So we have seen data sharing in terms of DDBJ, EMBL. They have their collaborative framework for sharing the genomic data. So this overall, the data in biological thermodynamic database is typically collected from variety of sources and is carefully curated and validated to ensure its accuracy and reliability. Okay, next we before starting going for discussing about how these mutations affect the binding or how these mutations will affect the stability. Let me discuss about the types of mutations. So we all know proteins are complex molecules made up of amino acids that play critical roles in various biological processes. Mutations in proteins can lead to changes in their structure and function, which can have significant consequences for the organism. Here are some types of protein mutations and examples. The first thing is missense mutation, so which involves single nucleotide change that results in a different amino acid being incorporated into protein. So these missense mutations can be neutral, harmful or beneficial depending on the location of the mutation and property of the new amino acid. Next is the nonsense mutation, so which create a premature stop codon that results in truncated or incomplete protein production. These nonsense mutations can lead to a loss of protein function and often associated with genetic disorders. The widely known example for this is the mutation from cysteine to alanine in CFTR, that is cystase fibrosis gene, causes the cystic fibrosis. Next is the frameshift mutation, which involves insertions or deletions of nucleotides, which causes a shift in the reading frame of a protein coding sequence. The frameshift mutations can lead to changes in amino acid sequence and loss of protein function. The famous example for frameshift mutation is sickle cell anemia, where insertion of a single nucleotide in HBB gene causes this, develops a truncated beta global protein, which leads to sickle cell anemia. Next is the silent mutations. These mutations involve a nucleotide change but does not involve an amino acid change due to degeneracy of a genetic code. So what is mean by degeneracy? So when we observe a codon table, a set of 3 codons or a set of 4 codons or 2 codons code for a similar amino acid. So that is what it means. We have a mutation, but even having a mutation, the codon still, the new codon still codes for the same amino acid. Where these kind of silent mutations are mostly observed in T53 gene, TP53 gene and all. And next is the splice site mutations. These occur at the junctions of introns and exons and can affect the splicing process of pmRNA into the mature RNA. So these splice site mutations can result in retention of intronic sequences and skipping of uh, exonic sequences and creation of novel splice sites. Generally, these are observed in muscular dystrophy. So these are some common types of mutations. So here, let me have some questions. So here I have some question for you. You can answer these questions using the chat box. So a mutation in a DNA can affect the amino acid sequence because it affects the code found in. So what mutation in DNA can affect so that the change we are seeing in amino acid sequence? I'll wait for a minute here. Answer. Yeah, here codons and non-codons. Uh, Codons are a set of amino acids. So when you see a exon and intron, they are the regions. And when we take a protein coding sequence, like an exon region, and this exon region is divided into a set of three. Like think we have an alphabetic like A B C D E F. So A B C is one codon, D E F is other codon. Yeah, yes. Coming to this solution, yeah. So the mutation in DNA will affect the mRNA, which mRNA is divided into set of three nucleotides, which are called as codons. So these codons will change so that amino acid, that particular codon will code for a new amino acid, causing a mutation. Yeah. Coming to the Q2, so when we have an uh, 11th amino acid would be serine. However, due to mutation, now it has amino acid alanine in that position. So what type of mutation could it have caused? Yeah, these are the mission mutations which code for, so which we observe a change in the codon, giving rise to a new amino acid. So here, because of the change, instead of serine, we are seeing here. Oh, sorry. Next, the final question is which type of mutation results in an incomplete polypeptide because of an early. Okay, I think people already started giving answers. Yeah. So the early access or the early observation of stop signal will give rise to a nonsense mutation. Yeah. So before moving on to studying the effects, I would like to take any questions on this mutation or till now what we have discussed. Okay, I'll move forward. Next is how these mutations will disrupt or affect the protein stability or protein interactions. So mutations can affect the stability in several ways, such as they can disrupt the delicate balance of interactions between amino acid residues in the protein, leading to changes in protein stability, folding, and function. Here are ways destabilizing mutations. So mutations that remove or introduce amino acids with unfavorable properties such as charged or bulky residues can destabilize the protein structure. These destabilizing mutations can disrupt the hydrogen bonds, salt bridges, and hydrophobic interactions, leading to unfolding of protein. For example, a glycine to aspartate mutation in a thermophilic enzyme, take a citrate synthase, destabilizes the protein and reduces its thermal stability. Next is the stabilizing mutations. So, same introduction. So, where they remove amino acids with favorable properties and can stabilize the protein structure. So, stabilizing mutations can enhance the hydrophobic interactions and promote folding of the protein. For example, substitution of glycine with proline in collagen stabilizes the triple helix structure of protein and prevents its degradation. Yeah, okay. I'll be discussing about point mutation after this slide. Next is the disulfide bond formation. So mutations that introduce or remove cysteine residues can affect the disulfide bond formation and stability of the protein. So this disulfide bond, for example, the substitution of cysteine with serine in insulin prevents the disulfide bond formation and leads to decreased protein stability and activity. Next is the conformational changes. Some mutations can also cause conformational changes in protein structure, leading to the altered stability. For example, a point mutation in P53 tumor suppressor protein can lead to the conformational changes that disrupts the protein stability and leads to cancer. So overall, the effect of mutations on protein stability depends on their specific location in the protein and resulting changes in the interaction between the amino acid residues. Understanding the effect of mutation on protein stability is crucial for predicting the impact of genetic variations on protein function and disease susceptibilities. Okay, I'll discuss about point mutation. See, generally we see point mutations means change of one. Here also the same thing, the concept behind the nonsense and missense, where the change of one nucleotide type or generally point mutation is a genetic mutation where single nucleotide base is changed, inserted or deleted in a DNA so that we see the effects in the downstream protein product. just like missense mutation, you see change of one, the amino acid starts coding for a new one. In nonsense, you see change of one nucleotide will code for a early stop codon instead of some amino acid. That's how. Yeah, they are part of missense mutations only. Yeah, and further, if we go for other classification of mutations, we have conservative, non-conservative, synonymous, and non-synonymous. Based on the effect they have, we have these mutations. Okay, the first question for today is, if free energies of a wild type and mutant are given, so what is the free energy change upon mutation? So consider free energy as a thermodynamic property or a value given. So what will be? Okay, B. E. Okay. Okay, I'll give you the formula here, where delta G here denotes for free energy. Okay, sorry, here the answer is B, not A. Uh, because when we take a wild type which is minus 16.3 kilocal per mole, and mutant as minus 15.2, when we do difference of these two, we get minus 1.1 as a stability value. And why the signs are important in thermodynamic analysis? So when you see a sign, a negative sign, considering a protein unfolding process, a negative sign. So when you denote a stability of a protein using a negative sign, that means we are telling that the process of unfolding is instantaneous. So the sign also has a crucial role. So the negative sign here denotes for this is a destabilizing mutation, that you see the protein total energy. So higher the total free energy, more stable the protein. So this mutation is reducing the free energy. So this is a destabilizing mutation. Okay, next is the, given the conservation scores of a residue. So what if the score is 1? What does it represent? And score is 9. What does it represent? uh, yeah, it could be anyways. It could depends on what your analysis also. When we take a folding experiment or a unfolding experiment, so generally the delta G is calculated by based on final state minus initial state. So it could be either ways also. Some softwares use mutant minus wild type, and others use. So the net value won't change. The sign will change. Yeah, I didn't check through the lectures. So if it is given in the lectures, we could have followed the two mutant minus wild type only. So here, yeah, score 1 represents the variable residues and score 9 represents the conservation. So this conservation of a score or conservation score refers to a degree of evolutionary conservation of its amino acid sequence across different species. So here is conservation score of 1 indicates the amino acid sequence is not at all conserved at that position, meaning that protein is highly variable across different organisms. On the other hand, a conservation score of 10 indicates that amino acid sequence is highly conserved and remain unchanged. I'm just giving the explanation of this answer only. Still, we are doing the second question as well. A second conservation of score. Yeah, moving on to the third one. So, name a web server for identifying the stabilizing center in protein structures. Now, okay, it's still showing one. Just give me a second. Sorry. Now it is moving. Okay. So here we are in the third question. So which server is used for identifying the stabilizing center in protein structures? Yes, KITE. So this KITE program is intended to select the stabilization centers, that is amino acids, involved in contacts which are important for maintaining the stability of a protein. So in this, two residues are defined as forming long range contact order. So that means, okay, in SKY, the stabilizing centers means residues involved in cooperative long range contacts. So here the cutoff for long range interactions is 10, but in general we use 12 also. Okay, moving on to the fourth one. So what will happen to the performance of protein stability prediction when the data set of mutations is classified based on secondary structure and solvent accessibility? Yes. Since secondary structure and solvent accessibility are important for identifying the protein stability, so these both classification and dataset will improve the performance. Next is stabilizing residues are mainly found in. Okay. Yeah. Stabilizing residues are mainly found in beta strands. So stabilizing residues in beta strands are typically hydrophobic and often located at the core of the protein. This is because the hydrophobic nature of these residues allows them to interact with the favorable other hydrophobic residues in the core and thus stabilizing the beta strand and overall protein structure. In addition, stabilizing residues in beta strand can also include polar and charged residues that participate in hydrogen bonding and electrostatic interactions with other residues in protein, further contributing to the stability of beta strands and as a protein whole. So next is a question for long range order. Even I think we solved this question in the last class. So again I want to repeat this question because it's an important concept in studying protein stability. So given a protein has 50 residues and 200 contacts, I know this is a time taking question, I'll wait. Yeah. So here is the formula for long range order where n denotes the binary value. So if the separation of two residues falling under it is more than 12, then we take n value as 1. So here you can only see a valine 71 and isoleucine 60 are the only residues that have a sequence separation more than 12. So two values. So 2 divided by total number of residues 50. So 2 divided by 50 gives this. Okay, moving on to the seventh question. If a glutamate is in a vicinity of lysine and is mutated to valine, so what will happen? Leave about what happens to the stability. So what will happen here? So what this mutation is removing from the protein structure? So here we are removing a charged residue, like we are replacing a charged residue with an hydrophobic or non-polar residue. So what will it cause? Yes, it causes removal of an ion pairs. So we are removing an ion pair. So when a polar or a charged amino acid is replaced with a non-polar one. Okay. Coming to the eighth question, which type of residues is dominant in stabilizing residues? Yeah, aliphatic residues or non-polar residues which form a stabilizing core. Next, given the free energy changes of alanine to alanine 12 to valine, alanine 24 to valine, alanine 25 to valine, alanine 20 to 28 to mutation. So, can you tell me what is the most tightly packed residue position before mutation? Yeah. Can anyone mention the reason as well? Okay. Here the value is P because the most tightly packed residues in a protein can identify based on this. Higher the delta G value, more tightly the packed residues in the protein structure, as mutating it would require the significant energy input to disrupt the surrounding interactions and packing. So here we have the surrounding hydrophobicity. I'll provide you with the formula as well. Okay. So here I was telling the higher the delta G value, the more tightly packed residues. How can I? Yeah, when you take an energy input, and here you see in a given question, we have different delta G values that have been given. So when you take a net value here, so the one having the highest is the A28P mutation. So in order to disrupt this, we need an energy input, so that since this is highly packed, to disrupting it need a higher energy input. So that is why this is more packing. And One more thing here is a sign. Positive sign corresponds to stabilizing mutation, and the negative sign corresponds to destabilizing mutation. So when we disturb the most tightly packed residue, it destabilizes the protein. So negative value and the net value both tells us here that A28, that alanine at 28th position is the mostly packed one. Okay, am I clear here? Uh, Generally tightly packed here means which falls into the hydrophobic core. Loosely packed, generally we see them on the surface. And if it is loosely packed, we don't see the much difference in their delta delta G values, even if you mutate them to other of similar type. Yeah, here when you take a residue, 327, it is surrounded by two alanines. So according to the formula, let's divide them into. So two alanines, so 2 into what is the hydrophobicity value of alanine. So according to a given value here, it is 1. So 2 into 1 is the hydrophobicity value added by alanines. Next we take valine. So 1 valine and hydrophobicity value is 2. So 2 plus 2, now we have 4. Next is 2 threonines. So the value is minus 1. So 4 minus, so 2 plus 2 minus 2. Next we have glutamine, where its hydrophobicity value is minus 2. So here again minus 4. Next is serine, which is minus 1. So because of 2 alanines, the hydrophobicity value is 2. 1 valine, the hydrophobicity value is 2 again. 2 threonines is minus 2. 2 glutamine, it's minus 4. And 1 serine, it's minus 1. So overall it leads to minus 3. The solution of these long range order questions and the surrounding hydrophobicity questions we have done in my previous video. So still, if anybody is having doubt, kindly watch the previous video. I couldn't solve them because I don't have my writing pad today. Okay, moving to the last part where I'll be providing the different databases. So first thing is Prothum. Coming to detail about Prothum, we have more than 1500 protein, 15,000. Now it's more than that. It's more than 30,000 we have now data in Prothum. So each entry in the database includes information on the protein sequence and the type of mutation and thermodynamic parameters of mutation, such as change in enthalpy, entropy, and experimental conditions under which mutation is performed. Next is ProNit, which has a data on the effect of single-site mutations on binding affinity of protein ligand complexes. Each entry in the database includes information on protein sequence, the type of mutations, the binding affinity of the complex before and after mutation, and experimental conditions under which the mutation was performed. Foldex database, this is built using the empirical force fields to predict the effect of mutations on protein stability. So this database contains information on thermodynamic and kinetic properties of thousands of protein mutants and can be used to predict the effect of mutations on protein folding, stability, and function. So there are several other databases as well. So these databases are useful resources for protein engineers, drug designers, and researchers interested in protein stability and structure function relationships. However, it is important to note that accuracy of predictions made based on these databases can vary depending on the specific protein and type of mutation being studied. Next is the prediction servers. So these tools are built to predict the effect of amino acid mutations or substitutions on stability. So they are developed based on different algorithms. Some are being used machine learning algorithms, some are being used physics based, some are being used observations based, some are being used deep learning based, everything. So some popular ones I'll be discussing. The first is the Foldex, so which is a widely used server for protein stability predictions. It uses empirical force fields to predict the change in free energy upon amino acid substitutions. And Foldex can also be used to calculate other structural properties such as binding affinity, protein-protein interactions, protein stability values. In Foldex, the protein stability is given as a split of different energy terms like contribution of hydrophobic interactions, contribution of hydrogen bonding, contribution of ionic interactions, contribution of van der Waals interactions, and contribution of entropy as well. Next is the Rosetta, which uses a combination of physics-based energy functions and statistical potentials to predict the change in free energy upon mutations. Rosetta can also be used for protein design and engineering. Next is a Duet. This is a machine learning model uses a support vector regression. So a regression algorithm to predict the delta delta G values and has been shown to have high accuracy in predicting protein stability changes. Next is a pop music, where it calculates the change in free energy of protein unfolding and refolding steps and uses them to predict delta. So it uses delta G to predict the delta delta G values. So this pop music can also be used to analyze the effect of mutations on protein dynamics and function. Next is STM, a site director mutator. This is a knowledge-based approach used to predict the effect of mutations on protein stability. So it uses a database of protein structures to calculate the frequency of different types of amino acid substitutions and their corresponding effects on protein stability. So STM can also be used to predict the effect of multiple mutations and to design new protein sequences with desired properties. Now there are several other methods as well. So these are just a few examples of the tool. Each server has its own strength and limitations, and the choice of the server would depend on the specific research question and protein system of interest. It is also important to validate the predictions using experimental methods. Even though there are several servers developed, still there is a need for more accurate server to match the experimental predictions. That's all for today. I can take any questions if you have. If no questions, you are free to leave the class. Thank you for attending. See you all on next Monday. Yeah, these free servers, yeah, are open source servers only, all of them. These are just few servers. There are some, I could tell nearly some hundreds of servers are available for globular protein stability analysis or binding analysis, developed by several groups across the world. Yeah, these are open source. You can also access them, and you can also access their published literature and review on that. And these open source softwares provide you with the data as well for your practice purpose or for your further analysis purpose. They provide you with a database these servers build on. Yeah, not website. You can search uh, some review papers that have been published in 2022. Exactly, I don't remember the authors of that, but there is some review papers. So you can, for example, you can type in Google Scholar or in PubMed, review articles on protein stability servers. You'll get the consolidated information, what is their performance, what is their advantages, limitation, drawbacks, everything in those review papers. I think 3 review papers have been published in 2022 on them. Like a Fang paper, he's published in 22, I think, by a review paper by Fang. He explained very well all these papers. Uh, what list you want me to give? I couldn't understand this question. What list you want me to give? Exactly the paper titles. Just give me a second. Uh, you can see in BMC bioinformatics, performance of web tools for predicting the changes in protein stability caused by mutation. In BMC bioinformatics by Anna Maraboti, it was published in 2021. The other thing will be, which is published in Briefings Bioinformatics in 2022, predicting the protein stability changes upon single point mutations, a thorough comparison of the available tools on dataset. This is by Piero. Okay, you can search BMC Bioinformatics and Briefings Bioinformatics for more paper, for review papers.